Hello, I'm Jan Leifman.
I'm Gayatri Pramal Menon.
Welcome to Med News Week Conference, where we feature presentations by medicine's global leaders. Today, we have an amazing keynote speaker in Dr. Jeff Karp. Dr. Jeff Karp is the Distinguished Chair in Clinical Anesthesiology at the Brigham and Women's Hospital.
In today's presentation, Dr. Karp discusses medical innovation.
Did you know that Dr. Karp is the co-founder of multiple internationally recognized companies? He's also the recipient of over 125 patents and 65 grants, with sponsorship by multiple companies, government agencies, and foundations, including the Bill and Melinda Gates Foundation and the NFL. He has also been recognized as one of 11 Boston doctors making medical breakthroughs by Boston Magazine. He's also recognized by MIT's Technology Review Magazine, or TR35, as one of the top innovators in the world. 3 members from his laboratory have subsequently received this award after him.
He truly is a pioneer in the field of medical innovation.
So let's tune in. Let's tune in and learn from this global leader.
Back in 2009, again, around the same time, Dr. Pedro Del Nido, who's the chief of cardiac surgery at Boston Children's Hospital, reached out to me. We had developed some adhesives, and he basically said that he was trying to treat children who have septal defects. So these are the holes in between the chambers of the heart. And he said, he goes to suture it sometimes and it just tears because it's so fragile. He said, there's these devices that work in adults, but you can't downsize them because the children's heart's, growing, and the child would just outgrow the device, and it's not acceptable to have to come back and do multiple revision procedures. So we thought about it and, quickly realized this is probably going to be one of the hardest projects we've ever done, because for an adhesive to work in this environment, it's just we're up against a lot of challenges. One is, it's very wet, clearly, a lot of cells and enzymes. You have all kinds of things. Every surface is covered with blood. So how are we going to get something to attach to the heart? We also, with the 60 beats per minute, normal kind of contractions, it's this expansion contraction. So it could, something could delaminate. And then we have the flow of the blood and that force. And again, that could cause delamination. So we kind of brainstormed and said, okay, if we could do this and it was to work well, what might that look like? And we thought, okay, well, if we could develop some sort of a patch that had a thin layer of glue on it, and we could put it into the heart and push it up around the surrounding tissue, and it could attach, and then cells could migrate on top of it to form new tissue, and then the material would degrade. The patient could be left with their own tissue sealing that hole, which could then naturally grow with the patient. So that's what we thought about, and we started to advance. And we quickly hit all kinds of walls, and seemed like in pretty much every project we get to the point where we're like, should we continue this? This really is not going well, and I think one of the challenges that we often face is that we approach something and we have expectations of what's going to happen, and then it doesn't happen. And then we step back and we rethink it, or we think that we're rethinking it, and we approach again, and it still doesn't work because we're really just applying the exact same thinking. And so I think one of the challenges for all of us is, how do we create tools to or use tools that can allow us to break free from this kind of conventional repetitive approach, where how do we really bring in new ideas to kind of hijack the way that our brains typically operate and the processes that we've kind of have become our habits. And so one of the ways that we do this in my laboratory, and there's many ways, I think, is to turn to nature for inspiration. So it's really this idea that every creature, every animal or plant that's alive today is here because it has solved an insurmountable number of problems. And those that haven't have quickly become extinct. So nature is truly the best problem solver. Hundreds of millions of years of research and development happening all around us. And there's lots of papers actually in the literature describing mechanisms for how things work in nature. And I just think it's so exciting. I think we're losing our connection to nature, and this is such an exciting way to start to regain it, is to start to, go into the academic literature or go for a walk in nature and just start to think about, okay, what solutions might exist here? What a great way to sort of leverage or harness our curiosity to bring in fresh ideas. And so we asked in this for this problem which we were facing, we said, okay, well, what creatures exist within wet dynamic environments, and maybe what could we learn from them that we're not currently thinking about? So we looked at sandcastle worms that exist in the sea. They sit on rocks and waves hit them and they remain put. And then slugs and snails. Sometimes you'll see, a snail on a leaf and it's raining and it's not moving, it's not falling off, it's not getting washed away. Sometimes it's walking on the ground and there's this kind of goo behind it. And we started looking at, okay, well, what do these creatures have in common? And we realized that they have a couple things that are interesting. One is viscous secretions. So things that are viscous tend to stay put for a short amount of time. Like if you put honey on a table or the wall, it doesn't wash away right away. And we thought that might be interesting because if we develop a patch and we have a thin layer of glue, well, maybe we'll put a precursor for that glue, and it could be like a viscous material. And that we put down, maybe we can get it to just stay long enough so the clinician can move it around to get it to the right site. Because we spoke to a bunch of doctors who said sometimes that's a problem. Just, if it's too adhesive and it just sticks, you want to be able to kind of move it around. We had been developing a lot of light activatable materials in the lab. And so we thought maybe we'll use light to get on-demand adhesion. The other thing that was interesting that these creatures had in common was those secretions contain hydrophobic agents, and things that are hydrophobic repel water. And so that immediately gave us an idea, because we had been thinking all along that we were going to perhaps use something that's hydrophilic, because to mimic the properties of tissues in the body, a lot of water content. But we thought, okay, well, what if we make this hydrophobic? It could repel the blood away from the surface of the heart. And this would allow us to get intimate contact. So all this was really interesting, but still didn't give us a sense of how we're going to make this thing super adhesive. So what we did was kind of like what ivy does, and that is, I don't know if anybody's ever tried to pull ivy from a surface, but it's incredibly difficult. It's really strongly attached. And the mechanism is that ivy has these root hairs that go up and down the surface, and they look for crevices, and then they secrete this kind of glue, and then it dries and they shrivel up. And you get this kind of mechanical interlocking of the glue and this kind of root hair. And so we thought, okay, well, if the glue could infiltrate into the tissue, light can go in maybe tens of microns, maybe 50 microns or so, maybe 100. We might be able to then cure the glue in the tissue and get this almost like kind of mechanical Velcro-like interaction. And so, and that's exactly what we found. I mean, after I'm sort of sparing you all the trial and error that we did to get this to happen. It took several months, but eventually on the top here, we basically just took the glue, the adhesive, put it onto the heart. We then immediately cured it with the light, freeze fractured it. And here you can see the collagen fibers. And you can see the glue's gone in between the collagen fibers here, and it's cured. So now it's like really strongly attached. So I'm going to show you one of the most challenging experiments we ever performed in the lab. This is an animal experiment. There is some blood here. I think this audience is probably used to seeing this, but if anyone isn't, just wanted to warn you in advance. But what we wanted to do here was see if we could seal a hole in a rat heart, actually a relatively large hole. So this is a dermal biopsy punch that we're using to create a hole. And we have this purse string suture here, so the animals don't bleed out during the procedure. We're going to remove that in a moment. And so we had to develop not only the glue, but also a patch. And the patch needed to be biodegradable, transparent, and elastic. So here we are. We put the patch onto the heart. There's a glue on the underside. The glue is light activatable. In this particular animal, something went wrong, actually quite wrong, which was one of the first animals we operated on. The patch was not big enough. It kind of slipped, so it attached, but it just didn't cover the full hole. So what we did was we had to sort of act quickly. We removed the suture, we took the glue, and we just put it on, just hoping for the best. And if you look carefully, you'll see that it's kind of less red where we have the glue. So it's repelling the blood away because it's hydrophobic. Because it's viscous, it stays in place long enough for another pulse of light. And we end up with a perfect seal. But we took these animals out 6 months and they all did fine. So this was really a great advance for us in the lab. Still a long way to go, but did give us a lot of energy and momentum to continue. So then what we did is we moved to a pig model, which was important for us to move to a large animal model, to pressure test, to really pressure test this, and then also to ask, could this attach inside a beating heart, exactly where you would have these septal defects. So Dr. Del Nido developed this cardioport device. We made an incision here in the myocardium, pushed this up against the septum, shined the light, removed it, sutured the myocardium. And here you can see two pigs, one on the left, here's the patch, and then the one on the right, here's the patch, 62 beats per minute. So I'll play that just one more time because it's a little quick. So here's the patch here on the left, and here's the one on the right, or was it 82 beats per minute? And then what we did is after 4 hours, we added epinephrine to increase the heart rate to see, okay, would this still, to really pressure test it more. Heart rate went up to 165, and you can see the patch here and the patch here. No sutures, no staples. This was just the glue and the patch. So after 24 hours, still a relatively short time point, but a lot of kind of beats of the heart in that time. We took a look. Here you can see a suture. This is part of the deployment mechanism, but the patch was indeed attached. And just to pause here for a moment, the two people who led this project in the lab, Nora, a cardiac surgeon from Germany, and Maria, material scientist in my laboratory. And Nora was in Dr. Del Nido's laboratory. And Nora and Maria forged this incredible collaboration, had this really great synergy, a surgeon and a material scientist. Initially I think there was some friction in terms of trying to figure out how to work together, but eventually they just thrived through this kind of multidisciplinary interaction, learning from each other, and really I think it's a big reason why we were able to advance this as far as we did. I don't have time to show you all the details, but we did also develop a way to deploy this via interventional approach. So a device that goes into the heart. It's a double balloon structure. So you basically put the catheter through the septal defect, and you have balloon on either side. And then the patch comes out, and the balloons push the patch up against the heart. And then you shine the light, and the balloon has a reflective coating. So the light bounces backwards through the patch, activates the glue, and then we can pull the catheter through a tiny hole that we leave in the patch, which then can self-seal. So we actually developed that just as a potential way that we might be able to deploy this via an interventional procedure. So we still continue to advance this work. The glue itself proved to be very interesting because we could seal the carotid artery of a pig, the aorta of a pig, and other tissues. So we started this company, Tisium, that I spoke briefly about a moment ago, and they received regulatory approval in Europe for vascular reconstruction, just for the glue, so sealing blood vessels. Now they're currently advancing to do sutureless nerve reconstruction. So the clinical trial just started, and tackless hernia repair. And so when we started the company, we quickly realized we need to develop devices to deploy this in the body. So here you can see we're spraying it onto tissue here. I've added a blue dye so the surgeon can see where it's placed. Here is in a water tank with a vacuum assisted device. So you can see it, it adheres really nicely underwater. And then we shine the light here on the left. You can see this is an LED light. And then here on the right, you can see that we can place this into very small environments via minimally invasive procedures into the body. And here's the manufacturing operation, which is outside of Paris, to do all the GMP manufacturing of the glue. And there were a lot of challenges in getting that to work. Didn't work initially. So we actually had to change a bunch of things. And then finally, I just wanted to tell you about another sort of offshoot of this story, which was I was talking about this work at the New England Aquarium at this really incredible conference called Zubiquity, where animal doctors and human doctors get together and compare notes. And there's actually the penguin exhibit at the New England Aquarium. Some people may recognize it. And here I am speaking, and the penguins are actually kind of not behaving, and they're super loud. And a little difficult to see because it's a bit washed out, but I had to keep stopping in this talk because the penguins were just going nuts. And I was afraid no one would hear what I was saying, but Dr. Bill Rosenblad did, and he's a doctor at Angel Memorial. So he is a veterinarian. And he was in charge of at the time, the oral and craniofacial medicine at this vet hospital. And he said, let's keep in touch. This might be useful for what we're doing someday. So he reached out to me a few months later, and he said he has a patient, which is a bulldog. He had pulled a tooth, and it left a hole from the oral to the nasal cavity, and it wasn't sealing. And he had done, I think, 3 tissue flaps where he cut tissue, pulled it over, sutured it, and it failed 3 separate times because there's just so many forces in the bulldog's mouth. So Dr. Rosenblatt said, okay, well, why don't we debride that hole? We'll put your glue in and we'll cure it multiple layers. So we'll kind of really build it up, and then we'll pull a tissue flap over top and suture it. And then maybe the material will provide some structural support. So instead of it all being on the tissue flap, it could be absorbed by the biomaterial, this cured glue, and then the glue will dissolve away, and eventually the tissue will just replace it. So here's what the hole looked like. This was a 10-year-old bulldog, was really in miserable condition, wasn't eating very well. You can see kind of part of the tissue flap that was still here, but mostly had kind of torn. And so what we did is we went in, we put the glue, multiple layers, hard to see, but it's here, it's a little shiny, and then redid the tissue flap, pulled that over, and sutured it. So the dog went away, came back a few weeks later. The previous tissue flaps had only lasted 2 or 3 days. And Dr. Rosenblad kind of was wrestling with the dog to get its mouth open, something some of you may be familiar with. I'm certainly am. And eventually he looked, and, it was a very kind of emotional moment because, he had known he had been treating this dog for years and years, and had 3 failed procedures, and it appeared here to have worked, and actually ended up working for a couple years, which we're all really excited about. And so the data that we were able to generate from this experiment, I want to show you in a before and after picture. So here is the bulldog before the procedure, and you can see he's extremely unhappy. And then here he is after the procedure, extremely happy. So we've gone from here, unhappy, to happy. The owner was really, excited about this. This actually, just to sort of state one more thing here, this really created a lot of energy in the lab because we'd never really thought that our work would apply to animals. We certainly use some animals in our experiments, and I think this to us is kind of maybe just a tiny, something to kind of give back. And so I did grand rounds at Angel Memorial, this animal hospital, and that's actually led to another collaboration. Currently, we're doing one of the technologies that we've been working on with a collaborator. We're looking at testing that in a cancer model in dogs. And so that's currently ongoing. But just sort of, I think to me, it was just fascinating how all of these technologies that we're developing, we think only, about human trials, that there may actually be this huge opportunity, especially where for animals, especially where the kind of current state of the art is just not good enough. And there may be opportunities to take even some experimental approaches to try them, because, sometimes they may just work. And then finally, I just wanted to share with you, I talked a lot about some of the technologies that we developed and kind of showed a lot of the good results. There certainly has been we've had our fair share of failures and challenges and sort of many moments where we just feel like, kind of thrown in the towel and this is just not working. And actually, one of those moments came to me when I was asked to speak at a TEDMed conference. I had memorized some speeches, like 2 or 3 minute speeches when I was younger. But most of these are like 15 or 20 minutes. I was kind of frightened about that, but I decided to go ahead because I just saw it as a challenge. Memorization's always been very challenging for me. So, spent a lot of time putting this talk together, practicing it over and over again. This was months of work, basically working on it every day. And then I showed up and they said, well, here's the controller to advance the slides. You can't advance it backwards. If you need to go backwards, you need to yell out, please reverse the slide. So I thought, okay, for sure, I'm never going to say that. I better not advance too rapidly. And they said, sometimes what happens is people freeze when they're on the stage. And they said, well, if that happens to you, don't run off the stage, because sometimes people do that. They said, just smile. So here's what happened to me. Let's see if I can play this here. By physician, so that we could study them and determine which drug killed the residual tumor. Rule number 3. Surround yourself by many examples in nature. So I stopped in the middle of my talk. I'd memorized the talk like so well. And as I was speaking, I was thinking about different things, and I then lost track of where I was. And I tried to do what they said. I tried to smile. I was like saying every swear word I knew in my mind, trying to figure out how I would get back. I then advanced the slide. It was a blank slide. I was like, oh my God, why is there a blank slide there? I advanced it again, realized the blank slide was a cue to myself, but then I realized I knew the lines to continue. And as I was headed off stage, they said to me, and this was very traumatic when it happened, I was like, they said to me, oh, we can just edit that out. And so online, if you look at this TEDMed talk I did, you won't see what I just showed you, because they actually have edited out this long, I don't know if it's like 15, 20-second pause or something like that. But I will say one more thing here, which is that experience actually has helped me improve my presentations, because I feel like I'm a lot more comfortable. The fact that I had stopped in front, there was 5 high definition cameras on me. It was really, it was at the Kennedy Center in Washington, D.C. And the fact that I was able to recover, that kind of actually gave me comfort kind of moving forward in my talks. And so I want to end with this quote, which is one of my favorite quotes of all time, by Michael Jordan, who says, "I've missed more than 9,000 shots in my career. I've lost almost 300 games. 26 times I've been trusted to take the game-winning shot and missed. I failed over and over and over again in my life, and that's why I succeed." And I think that it's the failure is it's the challenge is it's just so painful in the moment. But I've kind of realized over time, and it never goes away, it's always painful. But I think, as I was saying before, when we do fail, I look at it as an opportunity to be creative, to try to come up with, to try to learn something new, seek out some new insight, bring in somebody new to just re-energize the project. And almost every time it allows us to kind of figure out a new vertical to at least try. And sometimes that has, worked out really well. So at this time, I just want to thank everyone for your attention. I'm not sure if I can take questions here, but I'd be happy to do so. So thank you so much.